welcome to the Kids First Spring Public Webinar. And inspired on the picture you're seeing, I'm inviting you to join us to ride the wave of discovery supported by the Kids First program. Welcome to the Kids First Spring Public Webinar. Thank you for being here today and spending time with us. I'm Marcia Fournier. I'm a molecular biologist by education. I dedicated my career to precision medicine in the areas of oncology and rare diseases for the past 20 years. And I joined the program in 2022. I will be your master of ceremony today. You will hear from Dr. Helbig, one of our principal investigators. He will talk about Kids First supported research, and then followed by Dr. Higgins, who will join us to give a demo on the Kids First portal, and then talk about new advancements and technologies that will be available to the research community. I will start with a couple of housekeeping topics. Please use the Q&A box for your questions at any time of the event. We're going to be answering the questions during the event, as well as we're going to have a Q&A in the end. So with that, I will start with talking a little bit of the Kids First program history. The program was inspired on Gabriella Miller, and you see a picture of her and her mother, Ellen Miller, here. She was 10 years old when she passed from an aggressive brain cancer. And this program is part of her legacy, and Ellen Miller as well, carrying forward the mission of making this program to happen. The program was authorized by a bipartisan bill in 2014 and most recently renewed in January 2025. This program is a Deep KIPSI Common Fund program administered by 4 institutes at NIH, NICHD, NCI, NHLBI, and NHGRI. So Deep KIPSI and Common Fund are part of the NIH Office of Director. So the program aims to accelerate research with data sharing across pediatric conditions, including congenital anomalies and pediatric cancers. The program invests $12.6 million per year on data generation, data sharing, developing data resources. But more important, we are building a community around the data. A lot of people ask me, what type of research Kids First support? So Kids First supports data-driven research of congenital anomalies and childhood cancer. Data-driven research approaches are ways of doing science where the primary... sorry about that. I think the pool here disrupted my, so data-driven research is, let me just go back here. So data-driven research is where a question is done based on large amount of data, like the examples here, whole genomes of large cohorts to discover and prioritize novel variants. Epigenomic profiling, identifying regulatory mechanism, or long-read sequencing, uncovering structural variants. So these projects not only look at gene by gene and then link one gene to one disease, but they search broadly patterns across thousands of samples. So when I ask AI to read the Common Fund Kids First page, where we have abstract of projects that we support in the past 10 years. I asked AI to create a word cloud with the type of research we are supporting, and the type of scientific questions are questions about patient prognosis, patient treatments, finding novel biomarkers, patient risk assessment for patients, subtyping diseases, which often has an impact also on treatment decisions, and then causes of the conditions. But the big word you see here in the center of this word cloud is discovery, and that tells you the type of research we are enabling discoveries that will impact pediatric patients and families. Next slide, please. So other question I get is how I participate of this program. So if you're a patient, connect with a researcher. If you're a researcher and you're working with patient cohorts, you can come to NIH and respond to one of our annual calls and submit your proposal for data generation of your cohorts. So we do that once a year, and then we provide access to NIH genomic centers, genomic sequencing centers for generate the data. And once the data is ready, we fund a data resource center to compile the different data types that we generate. And then once NIH approves the quality of the data, this data goes back to the investigators. But then after 6 months exclusivity, this data is released publicly. So now the data is not only available to the original group, research group, but also to a broader research community through the Kids First portal. So next, the 3 milestones for us is when the data is ready, the data is delivered in the hand of the investigator, and then the big milestone when the data reach that bigger research community. Next slide. And next. So far, okay, can you go back? So far, we have supported 87 studies, and 36 studies have been released publicly. And that is through collaboration. In this picture is an example where we had NIH staff that is part of the Kids First working group. This is the group of NIH staff that oversees the program with investigators, our colleagues from Sequencing Center, from the Broad Institute, and the colleagues from the Data Resource Center. Next. So I'm going to give an example of one of the 87 studies so we can go through and understand how we go from data to impact. So this example, for instance, is the project from Dr. Adriana Letra on sequencing multiplex syndromic cleft lip palate families involving 828 participants and 168 families. So we not only have genomes of affected children, but also their families, which make this data even more powerful. So can you click the animation, please? So as I explained, this went through multiple milestones, and then next animation will show you the direct impact with once we release this study to a bigger research community, then I think one measurable impact is the users, secondary users, that now we're going to have access to that data. But we realize that for Kids First, the impact is not an endpoint. It is continuous. So the next animation will show that this project also won multiple new grants, and next animation will show this is also part of the impact because this is fostering more research, more grants means more research, and in this case for risk prediction and prevention, treatment, and strategies for these conditions. Next slide. So Kids First program is being funding. The majority of its fund goes to data generation with a great percentage of 69%. And then data resources, building resources that will make the data usable, and then also supporting researchers, secondary users to analyze the data. Next slide. So in summary, the Kids First continuum from data generation to real-world impact depends on key factors. One, generating multiple data types and being able, through our data resources, to make this data available and usable by the community. That community can find the data, use the data, interoperate with other platforms. And then also funding a community around the data, supporting data analysis and data use. And then that will lead to the discovery and impact, generating new knowledge and advances that will improve children's health. Next slide. So this slide is a big picture. So as I said, 36 studies already available. That means over 30,000 genomes of affected children and their families. And the graphic with the body parts shows all the studies that are available from head to toe. And these studies are also, please, I welcome you to explore the data. The QR code will take to the page where you can check all the data that is available, and you can explore it. And as a measure of impact, direct impact of making this data available publicly. And again, I want to emphasize not only that this original research group will have access to this data, but now a global research community can access this data. And the direct impact that we like to measure is people using the data. And then the graph on the right shows the data usage increase over time with a great momentum this year. So I hope you're going to be the next to explore and use Kids First data for your research. And next slide. Another way of measuring impact is taking a look of grant awards that are using Kids First data. So 14 NIH institutes and 1 CDC center have invested more than $240 million in using Kids First data resources or using a Kids First data set. And one thing I want to highlight on the left, where you have the amount invested in awards per institute, not only the institutes that are directly involved to Kids First, but also other institutes are granting awards using Kids First data. And on the right, I'm showing the types of grants that are being awarded, with research project grants being the majority, with our ones being the great majority of these 113 awards. And again, emphasize this is outside the Kids First funding. This is additional funding just because we're making that data available, building resources so make it easy for people to use the data, and then supporting the research community for analyzing the data. And that is the impact, the result impact. Next slide. The ultimate way of finding impact is through scientific publications. And Kids First publications showcase how the community is now advancing pediatric research. One thing I want to highlight is the areas of research that goes beyond the conditions we support, like congenital anomalies and pediatric cancers, also including papers on more basic biology and genomics, on technology and data science. Another thing I want to highlight is the impact areas where we see many publications on diagnostics and risk stratification, and then also therapeutic insights. And so I welcome you to check our publications. We have found already over 300 publications that are directly using Kids First data resources and data sets, and the QR code will take you there. And I think the result of this is really seeing an advance from, again, the data to a more precision medicine advancing children's health. Next slide. I selected few examples of publications, one on pediatric cancer, congenital anomaly, and cross conditions. So the first publications use Kids First data and resources, cloud resources, to advance new therapies for central nervous tumors. The second publication is a primary analysis of Kids First data together with other data to develop early screening for patients in congenital anomalies. And the third example is investigators combine Kids First data sets of congenital heart diseases and neuroblastoma to understand common developmental origins of these conditions. Sorry about that. So again, showing examples of the impact of Kids First research through emphasize that continuing from data to impact through investment in data generation, data resources, and supporting a community around the data to use the data and analyze the data. Next slide. So I hope I convinced you of this Kids First continuum, starting with the community power data resources and supporting research, and then seeing that as a result, new discoveries and impact, improving children's health. And I would like to thank our investigators for providing pictures of real kids participating, and for the families for consenting participation of the children on the research projects. Next slide. I'm already a little bit over, but I would like quickly to answer your question. How do I access this data? So Dr. Higgins actually will cover this in more details, but there are several ways you can interact with the data, starting with a quick visit to our website. And in the next slide, I want to call you also to check our grant opportunities. So I want to highlight that NIH now uses what is called highlighted topics to signal to the research community the priority areas that NIH is interested. And we would like to encourage you to explore these topics, and you actually can note alignment of your proposal to topics in your scientific aims page, and we encourage you to do so. We will also provide a link in the chat with some grant funding opportunities catered to the Kids First community. However, I also encourage you to go to the big pond and search active grants at the grants@nih.gov. And with that, I will open for questions. Please add your questions to the Q&A box, and we're going to be checking and answering. And because of time, I want to go right away to introduce our next speaker, Dr. Ingo Helbeck. And just give me a second here that I would like to properly introduce Dr. Helbeck with his Dr. Helbeck is a pediatric neurologist at the Children's Hospital of Philadelphia, specializing in epilepsy genetics. He's trained internationally across Germany, US, Australia. He has led major global research collaborations focused on identifying the genetic causes of epilepsy. Dr. Helbeck has contributed to the discovery of several key epilepsy-related genes, and currently leads efforts within the International League Against Epilepsy to advance genetic knowledge and literacy in the field. I always like to introduce my speakers with something that you're not going to find on their bio. So outside work, Ingo enjoys traveling with his family, cycling, writing for his blog Beyond the Ion Channel. Oh, I'm curious about that. And reading science fictions, especially the older classics. So welcome, Ingo, and I'm looking forward to your presentation. Thank you.
Thank you. And I hope I just subtly managed to change the screen so you can see my screen now, but the audio works fine. So I'm going to talk about some of our kind of Kids First supported research, especially with regards to genes of epilepsy. And I'm going to tell you in a minute how this is linked to the Kids First program. So the overall title of my team is going to be like genomes, phenomes, and time. I'm going to cover in these 20 minutes that I have 3 topics. I'm going to quickly introduce epilepsy genes. And then we go right into what I call phenotype science. Yeah, when we try to understand what is the difference between genomes and phenomes and how can our Kids First resources kind of help us with this. And in the end, I want to show you the impact that Marcia was talking about, the kind of what does this actually change in real life with our patients. This is a niche topic for probably many of you, but what has happened in the epilepsy field is that over the last 20 years, we have discovered hundreds of genetic causes. And this is one of our publications that is already a few years old. You can see this blue cloud just projecting upwards. On the top left, you can see some of our work with ClinGen, where we constantly kind of try to keep on top of which genes are actually true or not. And so we have an explanation in very severely affected people with epilepsy of almost 30%, so 1 in 3 patients. The challenge is that we have situations like this. So what I'm showing you here is a timeline of a single patient. The x-axis shows you the patient's age. And what is shown on the y-axis is seizure frequency. 5 is very high, 0 is seizure-free. And what we're looking at here is this first year of a child's life with a genetic epilepsy. And what we have been struggling with in child neurology, and this is true for many other pediatrics fields, pretty much any project within the Kids First world, is that sometimes these trajectories, these outcomes, are very unpredictable. So what you see on the left here, this first year of life, is the same gene, sometimes with the same variant, is compatible with 2 completely different outcomes. And this is the variability that is particularly prone in pediatric disorders. This is what distinguishes pediatric care from adult care, that we deal with lots of variability and lots of rare disorders. Here you can see that this initial part is consistent with one patient at the top who is becoming seizure-free for most of his life. And on the bottom, you can see a patient who has seizures on and off in most times. So our team has been involved traditionally with gene discovery in the epilepsy. So a very typical Kids First omics topic. And what we then decided is that we actually wanted to take a detour and think about how can we do phenotype science. So we would like to do precision medicine in the end, make sure that we have molecular treatments, disease-modifying therapies, but we felt that we needed to take this extra step and understand what is really happening with the clinical presentation and the phenotypes. And the reason why this has become an omics issue is because this is where omics is technically bad at. So we have a so-called phenotypic bottleneck. And I think especially when we think about Kids First and the cohorts kind of sequence with EXO-1, we're starting from a very good baseline of everybody having had genome sequencing. Yeah, we know that genomes can be generated at scale, 30,000 in Kids First. We know that the annotation is standardized. It's largely 1 or 2 dimensional. And when we think about a genome, we're actually not talking about the DNA in this person's body. We're talking about the digital representation that is on dbGaP that we can share and download. And this is what makes this so kind of catalytic as a factor for research. When we think about phenotypes, we have none of this. It needs to be manually curated, it's not standardized, and it's very sparse and highly dimensional. So what our team has worked with over the last almost like 10, 12 years is so-called phenotypic dictionaries. And they are actually quite simple. So a phenotypic dictionary, in this case, if you have a phenotypic dictionary that looks like a tree, it's called an ontology. And what you see here is the connection between different epilepsy phenotypes. Yeah, and I've chosen a few epilepsy phenotypes out of my field because they are very specific features that only make sense to a small group of clinicians. So if you showed me this for craniofacial anomalies or for brain tumors, I would be lost. I wouldn't quite know how to interpret them. But then I would be able to refocus and kind of make some sense of this because these terms are connected. And you can see it here, for example, that an early onset absence seizure is an absence seizure, is a subtype of a generalized onset seizure. So now you can imagine this dictionary for all possible conditions that a human or a child can have. And then you have the entirety of the what is called human phenotype ontology. We try to plot this here, and this, I don't know, sometimes I describe this, it looks like a spaghetti monster or like a fungus, but this is the connection of 16,000 clinical terms. And in the inside, you can see some of these seizure phenotypes that I mentioned earlier. So this is the entire connectivity of possible human or pediatric presentations or phenotypes across all domains. And this has made this easy right now for us to navigate through this with algorithms and computational tools like we use in genomics. So let me give you maybe a few insights how we can take this new knowledge and kind of work with this in a joint way where we look both at genomes or exomes and phenotypes and how we've applied this to our Kids First supported research. So first, when we think about phenotypes, there are actually different ways how we can do phenotyping. And this is a kind of modification of a slide that I typically show because people often don't agree what they actually would like to do. What I'm showing you here is there are different ways to think about this. So we can do this in a very detailed way at the bottom with very in-depth natural history studies where we kind of spend a day for a single patient to do clinical analyses. On the other hand, we can do very large-scale phenotyping on thousands of patients with just a few clinical terms. And the combination of this, or the middle ground, is what we call longitudinal phenotypic studies. And this is where I think lots of potential is right now, and which is what I'm going to focus on. Here, to take a step back, almost now 7 years ago, we kind of used this method initially to find genes. And this is the discovery of a gene that's called AP2M1, which is a brain gene associated with a severe epilepsy. And the reason or the way that we found this gene was actually a bit of like a coincidence or a, let's say, lucky opportunity at this point, because we discovered this gene through what we call computational phenotyping. So we used these clinical terms, these HPO terms, and we systematically compared them across each other within a cohort that was completely sequenced. This was a very small cohort. This was roughly 360 patients back then. But we compared every patient to each other using computational tools and asked the question, which patients are very clinically similar? And what we found that you can see here at the bottom, I'm going to mouse over this quickly, you can see this little distribution here. This is the distribution of what we call phenotypic similarities, so pairwise comparison between people. Then we found people with AP2M1 at the very end of this. So these people were very clinically similar, more than expected by chance. And this gave us a very strong evidence, and this has been confirmed since, that this is a disease gene that was found not through a high number of variants in a cohort, but having genetic variants and then having a very strong phenotypic resemblance, clinical similarity on top. From this, we've actually moved this on to quite a bit, and this is where this goes directly into some of the research that people are kind of starting with Kids First data now. We can do this at scale now. So here's one of our latest works where we've done this now on more than 11,000 people with neurodevelopmental disorders. This is work led by Shiva Ganesan in our lab. And what you see here is a demonstration of more than 670,000 phenotypic labels, and how they appear in this code and how we try to kind of make sense of them. And then if we do these kind of analyses trying to compare patients, we build very interesting networks. So this is now a comparison of 11,000 people with genome or exome sequencing with known variants linked by their phenotypes and asking the questions, how close are they to each other? Yeah. And without counting each and every dot, you can see that there are different colored spots and that some spots are very close to each other. Yeah, those are genes that we know that they kind of cluster quite clearly within phenotypic groups. And then you have other genes that are across the entire sphere. So they are somewhere in this kind of world where they're often quite different. Yeah? And so this is now a representation of an entire network of phenotypes, 11,000 patients, and all of these patients are kind of shown with links to each other. I promised that I would talk about time and our EMR-based research, and what you see here is one way how we've tried to understand how we can add yet another component to this that is very important for pediatric care. This is age. What you see here is what I call the tornado. Yeah? Most medical systems use a tool called Epic. Yeah? And at the time when we made this first analysis, Epic was exactly 10 years old. The x-axis is patient age, y-axis is patient stacked by their first encounter in the medical records. Every line is a single patient, every dot is a single patient encounter. So you're looking here at every neurology, brain tumor, epilepsy, headache note in our hospital, across more than 100,000 individuals. And this is 123,000 patient years. The reason why I'm showing this is because I would like to move away from the notion that rare disease is rare. Because what is really rare is our insights into this, but the data is quite ubiquitous. Neurology is composed of many rare disorders, and we have access to this data if we know how to approach it. This is our Kids First cohort. This is what we call the Kids First Neuroscience cohort, a combination of everybody who has brain tumors and epilepsy. Yeah? And you can see here we've overlapped this again. X-axis the age, y-axis are patients being stacked. In purple, that's the color of epilepsy, and blue are patients with brain tumors. And also here we're looking at almost 14,000 patient years, and every single of these individuals has a genome that we can look into and that we can mine. Those are here in this case roughly 2,500 people. So what can we use this for? What are the insights that we can get from these types of data? Well, we need to just understand first what we can see in there. I already introduced the HPO, the Human Phenotype Ontology. And what we did a few years ago is that we took our entire medical records, so data on more than 1.5 million patients, and translated this all into these terms using natural language processing, or AI. Yeah? So we have this big pool now of translated notes that all make sense, that are all connected with regards to content. And what we can build out of this are timelines and phenotypic stacks. What I'm showing you here is kind of some idea on how we did this with a gene that was called SCN1A. This is a very common epilepsy gene. Yeah? And what you see at the bottom are different stacks. So those are 3 months increments. And for every monthly bin and for every patient, we have stacked the clinical features using HPO terms in this document. This is almost like building Lego bricks. Yeah? What we've done here now is that we said we can actually follow a phenotypic trajectory. We know when patients are diagnosed, and we have a good sense on what happens before, at the diagnosis, and what happens afterwards. This allows you to kind of get some very interesting insights. I'm going to show you quickly how we can move this towards medications and how we can use this for risk prediction. Again, here's our cohort. This is the Kids First Neuroscience cohort, brain tumors and epilepsy. And now we can overlay some of the methodologies that medical record research allows us to do. So here you can see again, the x-axis is patient age, the y-axis is the seizure frequency that I showed you earlier on my very first slides. And you can see how a patient goes up and down. And you've seen I've put 2 colors on this. In green, you see periods when someone has a reduction in seizure frequency. In the blue, you have periods of longer seizure freedom. And we can now combine this with another information on the medical records. And this is the type of epilepsy medication that a child gets. Yeah? And these kind of abbreviations here stand for common seizure medications. And now we can compare the medications with what is actually happening in real life. And people call this real world data in our patient cohort. Those are the medications prescribed over time. And now we're going to take it 3 conditions that most of you will never have heard of. This is 3 of these kind of many rare disorders that we have in child neurology. Again, pediatrics, and especially the conditions that we assemble in Kids First, are often very rare conditions. And those are now 3 epilepsy conditions. I already mentioned SCN1A. The different genes are called SCN8A and SCN2A, and then SDXBP1. And what you see here is a so-called medication landscape. The colors are always the same, but it shows you the different medications that these patients have had over the lifespan, or this cohort had. We can now use the medication and response, look at the seizures over time, those are seizure plots for these 3 different genes, and then ask the question, what medication reduces seizures at what time, and is there any difference in how these patients respond? And what is quite interesting is that even in these cohorts that are still relatively small, 20 patients, 15 patients, we see sometimes very strong patterns. I'm showing you two here. Those are two conditions, SCN8A and STXBP1, two of these kind of many rare epilepsies that we have out there. The genetic conditions are rare. Those are two of them. And here we look at these therapies and we compare medications across each other from existing data in the cohort. And I've highlighted two lines here. There's a medication OXC is oxcarbazepine, which is an epilepsy medication, and keto is the ketogenic diet, a dietary therapy. And you can see that there's almost an opposite effect when it comes to the response to these two medications. Oxcarbazepine works better in SCN8A versus ketogenic diet, which works better in people with STXBP1. So using this data, having genomic data, and having access to medical record data allows us for some very, very key insights for medication response that we can use clinically in these patient cohorts going forward. And you would imagine assembling this data with prospective studies is nearly impossible. It is kind of almost impossible to systematically follow patients long enough with a rare disease to have these particular types of insights. So we can do something else, and then we're kind of going a bit into like omics and large-scale data analysis. Here we're looking at the SCN1A gene again. And this is now a cohort that looks at roughly 30 patients. And what you see at the top, again, this is the x-axis is patient age. The y-axis looks at frequency. And we're looking at the frequency of a very particular dangerous phenotype, which is called status epilepticus. So this is a seizure that is very long. This is a seizure type that many of our brain tumor patients also have. And it shows you the frequency of status epilepticus in these patients with SCN1A and everybody else in gray. And you can see there's a dramatic difference. We can see a frequency of up to 50% in the first 2 years of life. And then what we can build out of these distributions is what we call fingerprints. And this is what you can see at the bottom. We've taken the peak of the difference for every single phenotype and assembled this kind of footprint at the bottom that is actually quite unique for each specific gene. So we're going in, we have a rare disease that we know very little about on how it actually does in our kind of ongoing cohort. Many of the studies are old, are kind of historic, but here we have actual data, real world data that shows us how the fingerprint of a certain condition is. And then you can even move forward. And this was kind of a paper that we published 2 years ago. That was one of the kind of papers of the year in genetics and medicine. We can now use this and say, let's forget about the genetic testing. Let's take every single patient note that is written after genetic testing and just ignore this because we don't want to be biased. And then we can ask the question, how good are we using this data to predict a genetic cause from clinical data alone through phenotypic data? So the routine data that is being assembled in the emergency rooms, in the hospitals, in an outpatient setting, how good is this? And for this particular gene, as an example again, we have an accuracy of over 90% to identify this genetic cause based on clinical data alone, more than 6 months before the genetic diagnosis was made. And those are not sophisticated tools. There's no AI in there. There's a simple machine learning. We've kind of built out this little panel here in D, which is a decision tree that is data-driven. And this gives us the idea that using these methods, we can come to very, very clear and very practically helpful insights into how we can make a diagnosis way before we have genetic testing. So I wanted to give you a brief insight into what we do with genomes plus phenomes. Again, our focus is on kind of using resources such as Kids First, where we have genetic data and kind of take this additional step and look into phenotypes. Epilepsy genetics in itself is like a long success story, and we try to understand how can we translate this now into treatment insights. I talked about phenotype science, and then our current applications, how we can think about medication response and how we can predict genetic diagnosis way ahead of the actual genetic testing. Yeah, here's an overview of my lab. I worked at Children's Hospital Philadelphia in neurology, and thank you so much for giving me a chance to talk here today. And I think, Marcia, this is back to you now. Yeah.
Getting not the claps, Ingo.
Yes, I lost my screen for just a second, but yes, I see. I just saw the virtual claps. Thank you.
Excellent. Thank you so much, Ingo. Great. Well, I can go ahead and, oh, hey, Marcia.
Hello. I was going to give you a quick introduction, but I think you can give it faster because you
Sure.
are chasing your trawler,
I
chasing
am.
sidewalks and through your neighborhood, so you're going to be able to chase.
I am. Thank you so much. Yes, let me find the right share button here and let's dive in. Yes. So thank you so much, Marcia. My name is David Higgins. I work at the Kids First DRC, and I'm so excited to give you an update about what we've been working on here at the Kids First DRC. Ours is going to be framed a little bit around what we've been doing, what you can do today, and what we can do in the summer. And as Marcia said, my fun fact is that I have a very active 2.5-year-old, so I'm always moving. So we're going to keep things in that theme. We're going to keep things moving quickly here. So what have we been up to here at Kids First DRC? So over the last year, we've been moving. So not physically, we're still at the same institutions, but we have moved the data. So we've migrated our entire data set from one storage location to another storage location. So analogous to moving your house, you might have packed things up into a big truck. We didn't have to haul any boxes onto a trucks, but we were accounting for millions of files, which represent petabytes of data. And so lots of kind of lists and files and making sure everything is that is packed up, gets unpacked properly in its storage location. We are just about done, just wrapping up some final access issues with a handful of studies. But if you have any continuing issues following our move, please let us know and reach out to our help desk. We can get back to you. We've also been building new tools and workflows. So internally, we've been redesigning our database, which allows us to bring in clinical data from investigators, submitters like Dr. Helbig. And so this won't appear immediately. There's no website that you'll be able to click on and see. But in the back end, this will allow us to be able to receive, review, and release data more rapidly with faster and more comprehensive mapping to our ontologies, which Ingo was showing. So there will be improved data in the future and data will get out more rapidly. So we're excited to roll that out. So that's what we've been up to the last couple of months. But I want to talk about things that are going on today. What can you do now with Kids First data? And so I want to build on, so Marcia mentioned 3 recent publications, and I actually want to highlight one of those 3 that she picked out, evaluating the genetic overlap between congenital heart disease and neuroblastoma risk. And so I was really excited when this paper came out because the whole mission of Kids First is to find not just the genetic causes of these individual pediatric conditions, these individual congenital anomalies and individual pediatric cancers, but the overlap, kind of that, the middle of the Venn diagram here of what common pathways, what common genes might be causing multiple of these different seemingly disparate conditions. And so that's why I was so excited about this one, because it really kind of dials into that sweet spot in the middle. I'm going to show a little bit more details here. So what they have here is they looked at two Kids First cohorts, the congenital heart disease cohort and the neuroblastoma cohort. And then they rolled up to the gene level and they looked for enrichments in genes, variants within genes inside of each of those individual cohorts. And so along the x-axis here, this would be enrichment in the neuroblastoma cohort. So these genes are more implicated in neuroblastoma. And then along the y-axis here, this was the heart disease cohort. So these genes would be more implicated in heart disease. And so what they found is that there's 7 genes, this cluster right here in the middle, with increased enrichment in both cohorts. Kind of this, again, highlighting this potential shared overlap between these 2 different conditions. So this is the same. So this is the 7 genes that have been pulled out into a table view, which provides some additional description of the particular diagnoses that these patients were diagnosed with in terms of kids in the heart defect cohort that are carrying variants in these particular genes. And so what we can do, we can kind of take this the next step further here on the Kids First portal. So we could take one of these genes, this gene IREB2, which has been listed by the authors as a gene with SNPs that are enriched in both of these two different cohorts. Is there any evidence of various variants in this gene in other Kids First cohorts? Could be a follow-up question. And so we can actually really rapidly search for that here on the Kids First portal, which I'll do for my demo today. So I'm going to exit out of my slides, and I'm going to navigate to our portal website, portal.kidsfirstdrc.org. That will take you to this page. I don't have an account, so I'm going to log in, but if you need to make an account, it takes minutes to sign up very, very rapidly. I'm going to log in with my NIH ERA Commons ID, which I have. If you don't have an ERA Commons ID, you can log in with any Google authenticated email address or an ORC ID are your other options to log into our portal. I'm going to include my name and password. And as it loads up, I'm in. So I'm going to jump past the studies page. The studies includes information about our studies, data exploration, as information about participants, as well as our specimens and genomic files. But today I'm going to look in the variants tab and look on the germline variants. And so we can recall back to our demo here. We're interested in our specific gene we're going to look up today is IREB2. And so we can actually search this record of 430 million genetic variants in the collection of all the Kids First studies. We can search for that and try to find interesting variants for us. So if we want to follow up on this paper, I can go into the search here and I can search by gene name. So I can search IREB2 and hit enter. And so that pulls up. So within Kids First, there are 11,000 variants in the gene IREB2. So many of these, so they're sorted by levels of deleterious. And so the most deleterious are listed first. So this would also include silent mutations too. But we can then modify this in order to highlight just the ones that might be causing a phenotype or an effect on a participant. So we can filter by pathogenicity. So we have a couple different filter options that you can apply, but all of our variants are VEP annotated. So we can go into VEP and choose ones that have been scored VEP high and click OK. And so this gives us 21 variants that are in this gene that have a high effect. And so the analysis is a little bit imperfect to our paper. So the paper, they rolled up to the gene level, whereas here on the Kids First portal, we're looking at the variant level of each individual variant listed within this gene. But for the sake of example, we can click on this first one here, and it brings us to this variant page where it tells us about this specific mutation. It's a splice donor mutation, so it disrupts splicing, which causes significant effects on the function of the protein. It tells us that there are 14 participants of the almost 24,000 participants on the portal have this specific variant, not just the variant in this gene, but this specific variant. And as well as it's annotated with the nomad allele frequency as well to give us some additional context. So if we scroll down, it gives us information about what studies this variant is found in. And so we can actually see that, there's one participant in the neuroblastoma study and 2 participants in the heart defect study that have this specific variant in IREB2. But if you look more broadly, you can see it's actually present in other studies as well. So kids in the diaphragmatic hernia study, the orofacial cleft study, the kidney defect study as well also have this mutation in addition to that. So it gives kind of additional kind of context and shows that, we're going to be able to expand beyond this initial search and begin to kind of see what other information is out there. So if we then wanted to apply for access to these studies, we could then click on the links and it takes us to data exploration where we can search the participants in more details. And then we can link us all the way out to dbGaP where we could apply to receive access to the genomic data. And I'll let that load up real quick. So that's my quick little recap of what's possible here today on the portal. And as I'll allow that to load, but I'm going to return to my slides here. So I do want to highlight what we're up to now and what to expect coming up soon from Kids First DRC. So there's our boy on the surfboard getting ready for summer here in early May. We're coming up on Memorial Day. It's almost summertime for us. So he's probably on summer break. The Kids First DRC is not going to be on summer break. We've got a lot of exciting things that we're going to be rolling out this summer for all of our users to kind of continue the exploration and research using Kids First data. So what's coming this summer? New genomic data. So we're going to be releasing a bunch of data this summer, which I've kind of categorized under 4 different categories. So we're going to have some entirely new studies on the portal, including a rhabdomyosarcoma study, as well as a children's oncology group pan-cancer study from the cancer side. And then we're going to have 4 new congenital anomaly studies as well, one focused on hearing and deafness, a vascular anomaly study, a Hirschsprung's disease study, and a clubfoot study. So 6 entirely new studies on the portal. And then in addition to that, we're going to be greatly expanding a large number of our existing studies as well. So these are current studies that are already up that will now be adding more participants, more samples, more genomes to. So we're very excited to get all this genomic data out onto the portal and into your hands so that way you can use it for exciting research. So look forward to that this summer. We're going to be improving our clinical data too. So we've been partnering with research investigator groups, especially the orofacial cleft group and the chromosome 18 group, who've submitted additional clinical data about the participants that are already released on the study. So we're going to be updating that information to have more accurate, more specific terms for the ontologies that can better describe their conditions that they are affected by. So look forward to that coming this summer. We're also going to be releasing new genomic data files. So we've been reprocessing all the Kids First germline samples from the entire history of Kids First with a new Dragon-based workflow. If you're not familiar with Dragon, Dragon is a new bioinformatics suite of software that has a lot of advantages over the existing GATK-based software that a lot of Kids First data is based on now, including both a faster processing time as well as more accurate variant calling. So we're excited to get this new data out on the portal and improved data files from what's currently available. Furthermore, we're also releasing additional long read cohorts, expanding on the long read options that are already available. And we're going to be releasing brand new methylation data, which will be the first of its kind for Kids First. So exciting to get that out too. We're going to continue to be available. So as these new things come out, if you have questions, you can always stop by one of our monthly help sessions. We offer them twice a month. The first Tuesday of the month is Support You, which is for new users. It's at 10 a.m. Eastern Time. And then the third Thursday of the month is our masterclass for more advanced users. That's at 2 p.m. Thursday afternoons. If one time works for you, then just show up to that one. Even if you're a new user and only available Thursdays, we're happy to have anyone at any of our sessions. So please stop by, and there's details on our website for more information of how to join. And then I'll also highlight the CFDE Decode Institute. So this is a summer-long training program in data science. And so what is really exciting from our partners in CFDE, the Common Fund Data Ecosystem Training Center. So Kids First is part of the CFDE organization. And so we're excited to support their summer training sessions that they're hosting. And so this is a really exciting format because you can pick and choose specific sessions to kind of guide your own learning. I think it starts in early June and runs through late July, but you don't have to go to every session. You can choose the ones that are most interesting and exciting to you. And so DRC will be presenting and participating in a session on genomic data and how to combine Kids First data sets with other Common Fund data sets. And best of all, they're free to attend and sign up. So if you're looking to hone your skills or learn some new skills this summer, check out the CFDE Decode Institute. And so that is it for me. So we are going to rapidly get back to work here at the Kids First DRC. We do have a couple minutes for Q&A, which I'll pause for. But before I do that, 2 final announcements. First, please take the post-event survey. So when the webinar ends today, a link will pop up taking you to our survey. Please take a moment to fill it out. It helps us plan for the future events and drive our agendas that we're setting. So please take a moment to provide feedback or our event today. And then also save the date for September 17th. We'll be back here for the fall webinar, telling you all about what we did this summer and all the exciting things that we've got going on at Kids First. And so with that, I think we've got about 4 minutes to spare before the top of the hour here. And so I will stop my sharing and see if there's any final questions or thoughts before we wrap up.
Thank you both, David and Ingo. This was amazing. I have a question here for Ingo. Dr. Helbeck, for congenital birth defects with limit phenotype variations, how can we apply HPO to predict genetic and environmental causes?
So with limited phenotype variation, maybe, Walid, would you mind specifying what you mean by limited phenotype variation? Or maybe I can maybe just kind of see how I would think about this. So we have, so congenital birth defects are typically diagnosed at birth, given the features that we see. And I think the challenge that we have in terms of making diagnosis is, first of all, kind of, can we shorten the time to diagnosis, and is there anything we can add in specifically with regards to phenotypes? In our field in neurology, we're already kind of having quite a battle because we're dealing with brain phenotypes that are not really like hard-coded in someone's appearance or are a bit more obvious. Many of the clinical features that are being used actually in congenital birth defects are very strong, and there are some very good prediction tools already out there. There was a tool coming up a few months ago that was published in Nature that's called, was a foundational model for rare disease. And the way that you can think about this is that phenotypic features have a certain value. And the more specific and the less common features, so the more specific or the more valuable this is. And many of the HPO tools use what is called information content. So if you have a feature that's very rare, it's very significant, it kind of drives the discovery because it's so informative.
Thank you. Any additional questions? Oh, I have a question for both you, David, and Hingo. I think that I get questions from a lot of people that haven't used yet Kids First data. David, per perspective of who is like receiving the questions, and Ingo, because you use Kids First data, what were the big challenges? And then what was easy for you, like, to start interacting with our data?
Yeah, I can kind of feel that. So I would say from working with, I would say from working with users, I think we get questions about connecting the portal, and so or applying for access. So all of our Kids First studies are available through dbGaP. There is each individual you can apply, but you can do one application to get all the studies at once if you're interested. And so kind of that connection of, navigating the different portals, so then you can work from dbGaP to get access, find the files on Kids First, and then do the analysis in Cavatica. And so a meeting with users and kind of, that's one of these things that we talk about on office hours, is kind of guiding them through this research journey of how you would start and then how you could find what you're looking for, and then now you're ready to go off and explore. So that's something that we talk about and are happy to help with.
Yeah, and David, I would say maybe from the same thing, because I think I'm just going to use myself, so I don't need to talk about anyone else. Typically, you approach Kids First from a very naive standpoint, and of course, what every researcher always wants is kind of, can I download all the data? And then, like, Kids First is at this level, you can say it's actually no longer possible because this data set is so large. We've seen this in the cancer space that moved to cloud the earliest, and I think this is where probably many investigators need a bit of like a rethink to say we need to shift our entire idea on how we would like to have the data somewhere, because we cannot just say, give me all the data and I'll do this myself. This is where it's actually good in the pediatric field to have this cohort where these problems arise.
Absolutely. Yeah, there's so many participants, so many samples that, the individual computers are run out of room, and so having the shared cloud space is a great opportunity to teach folks.
Well, we are on top of the hour, and so it's time to say goodbye to everybody. But this should be seen just as a beginning. I think the webinar goal is for you to get the first, get to know the program, some of the participants, some of our research, and I really appreciate David and Ingo to be here today, and all of you attending the webinar. So feel free to continue, like our continue from data to impact also involve this engagement. So I would like to invite you to engage with us after this webinar through the different forums that we host, and also through our website and emails. So thank you again, and I'll see you in the fall webinar, and have happy summer, everybody.
Thank you, Marcia. Thanks, everyone.